Okay, so here we are back again, continuing our investigation through the immune system. Theory, we'll get there. Okay. And where we're going to start off today is with the adaptive defenses. And again, this is specific, right? This is one exact pathogen. This is H1N1, SARS, SARS-CoV-19, whatever. Also, abnormal body cells. So anything that's been virally infected, obviously, falls under that bin. Cancer cells, stuff like that. Now, again, we're not unique here. We're not all by ourselves. We have lots of interactions with the innate defenses we talked about before, right? Particularly as we talk about inflammation, but also if you remember when we talked about complement, right? That classical pathway of activation requires an antibody to serve kind of as a chaperone. Antibodies, of course, are part of this adaptive defense, and so it doesn't happen without an adaptive response. The beauty of the adaptive defense is that, of course, it can be highly effective, but there's a couple of caveats to it, right? We have to actually have an infection in place before we can begin doing it, right? We have to have the pathogen inside of our body before we can even begin to mount the response. So that's kind of a bummer, right? And then once we do begin to start this response, it can take time, right? It's actually pretty slow. And really, honestly, on top of that, kind of building on the idea that it's slow, is that the first response that we've got kind of sucks, right? The adaptive defense is really only going to shine when we have kind of subsequent infections, right? So it's something we've gotten before, and then we've got it again, right? And so that's kind of the idea behind vaccines, which we'll talk about, or inoculations too, which we'll talk about a little bit later on. So as far as the adaptive defense, itself goes, again, specific, right? Very, very specific things. It's going to look for something we'll talk about called antigens. It's systemic, right? The innate immune system is very much about solving a problem in one spot, right? Inflammation is local, right? Fever is body-wide, but inflammation and most of those responses are local, right? Systemic here means that we're going to dump this stuff out in the blood, and it can be anywhere and everywhere, right? It's not restricted to the initial site. And then most importantly here, where it's really going to shine is that we have a memory, right? We do better the second time, the third time, the fourth time around because we've learned something about what it is, and we have a better attack ready to go off the bat. We're going to divide our adaptive defenses up kind of in the same way we divided the innate defenses up into two parts. Here, the adaptive defenses parts are going to be based around what kind of lymphocyte we're going to be dealing with. So we have humoral immunity, which is antibody mediated. So this is the antibody, the B cell part of this. And then the cellular immunity, which is going to be the T cell half of this. Again, humoral immunity, right? All about lymphocyte, or B cells, antibodies, that kind of stuff. And it's almost a little passive in some sense, because an antibody doesn't actually do anything by itself necessarily. There are some things that it does do, but generally speaking, its big role is simply to opsonize, right? A term we saw back in the first video, or to mark, another way to put it, pathogens for destructions here by the innate defenses. Now, importantly here, the humoral and the cellular immunity branches actually don't interact with each other. A T cell does not look for an antibody. So very much going their separate ways here. And the antibodies are all about marking things off for the innate defenses. T cells here, the cellular immunity part of this, right? This is direct action, right? We're going out and we're actually killing stuff. So that's a little more proactive, maybe. But at the same time that we're doing this, we're also having lots of indirect actions here, particularly, again, I'm going to say this till I'm blue in the face, enhancing inflammation, right? There are also going to be certain kinds of T cells that are involved in activating other lymphocytes, okay, both T and B cells. We'll talk about what activating means here in a little bit. Okay. All of them, however, are keyed around the idea of this antigen. And we've talked about the term antigen before, right? Again, it's just a large, complex molecule, right, that basically just is a name tag, right? The adaptive responses, the antigens we're looking for here with the adaptive response are ones that are associated with non-self cells. And we saw kind of what happens with this kind of reaction when we looked at blood typing, right? We're looking for A or B antigens, right? And when we found them, we had a pretty profound reaction to it. As far as antigens themselves go, right, they are actually going to come in a couple of different forms. The ones we spend the most time worrying about, and we're talking about the most here, antigens anyway, are going to be what we call complete antigens. And a complete antigen is going to be something that causes an immune response by itself, right? It is immunogenic and reactive, right? Basically, what this means is it doesn't need any help, okay? So this is going to be, something that we associate with a bacteria, right? Or a virally infected cell or a cancer cell, anything that is foreign, right? Incomplete antigens, or things we're going to call haptens, require us to participate here a little bit. So not everybody is going to react to a hapten, right? Not everybody's going to have a response. These are going to be things that we typically associate with, say, allergies, right? So reactions to poison ivy, to pet dander, to detergents, to any of those kinds of things that can cause reactions in some people, but not everybody. These are going to be incomplete antigens or haptens. And so we don't worry a whole lot about them here, but they're going to operate more or less the same way that a complete antigen does in people that are susceptible to them. Now, when it comes to the antigen itself, the whole thing actually is not necessarily valuable or useful to us. It's really only a small part of it, right? And that little small part is what we call the antigenic determinant. And the antigenic determinant is where the antibody or the lymphocyte or whatever else actually binds to the antigen. So here in our little cartoon, right here in red, we have this big structure here with all these parts sticking out of it. This is the antigen. But the parts that are sticking out of it, right, this little triangle house looking one here, this roundy one, this middle finger guy here, right, these guys over here, these are the antigenic determinants. And so our antibodies, A, B, and C here, are going to bind not here to the antigen, but just to the determinant. And what this means for us is that if we're gonna try to prime a lymphocyte in some way to be responsive to an antigen, we don't have to have the whole thing. We only have to have a small part of it. The other part that's important here is that antigens typically have multiple different determinants. And so we can have multiple different antibodies all binding to the same antigen, all opsonizing the same antigen, which can help with an immune response for us. They're also present, of course, on the cells of your own body, right? We call these guys self-antigens. And these are what we talked about before when we talked about transfusion reactions, right? So if you get the cells from an organ or a graft or a transfusion from another person and the self-antigens are not compatible, this can cause you to have a rejection of those foreign tissues. These can be minor, this can be pretty severe. Generally speaking, they're bad, even when they're minor. When we talk about antigens, they're going to be held in these big proteins called major histocompatibility complexes, or MHCs. And the MHC has a little binding site on it that holds the antigen, at least the antigenic determinant. Okay, and that antigenic determinant can either be a self-antigen or a foreign one, depending on the type of MHC we're talking about. So the cells of your body, except for red blood cells, bear what are called MHC class 1s. Okay, and so these are going to hold self-antigens. MHC2s, on the other hand, hold foreign antigens, and they're only going to be present on certain kinds of immune cells for reasons that we'll talk about a little bit later on. Basically, the idea here is that they're going to play a role here in turning on and maturing lymphocytes to react to these foreign antigens. When it comes to cells here, right, two of these guys we're already familiar with, T cells and B cells, right? But the third kind, right, these antigen-presenting cells, APCs, these are where those MHC class II proteins are going to come into play, right? And so these guys are vitally important because without these guys, there is to be no activation of any of our lymphocytes. They are going to be a big step in making this stuff happen for us. So a lymphocyte itself is going to go through basically 5 stages of life, right? We've talked before, we talked about the circulatory system, about leukopoiesis here, this origin of them. They're all going to develop from hematopoietic stem cells in the red marrow, yada, yada, yada. From there, though, we're going to diverge, right? And we're going to go through a maturation, a seeding, an antigen encounter/activation stage, and a proliferation stage if the particular lymphocyte completes its life cycle. So maturation here, this is kind of that boarding school that we talked about before, right? And boarding school is going to exist in different places for different cells. So our B cells are going to have their boarding school in the bone marrow, right? T cells, remember, mature in the thymus, okay? And in the marrow of the thymus, wherever the school happens to be, they're going to be tested for 2 different things, right? And these 2 things add up to what we call immunocompetence. And basically, the idea is, are you competent to do the job, right? Can you actually do the thing we ask you to do without totally screwing something else up? So for these 2 different types of cells, there are different meanings behind immunocompetence. So for a T cell, it must be able to recognize and bind to a self receptor, right? It has to be able to bind to a MHC class I and bind to it, otherwise it can't do its job. B cells, on the other hand, have to be able to produce a receptor protein, right? And again, if they can't do that, they can't do their job. Once they've managed to do that, right, then they move on to, are they self-tolerant? Can the lymphocyte recognize cells that belong to your body and leave them the hell alone, right? If they fail either of these 2 steps, they get destroyed, right? Because they're either incompetent, they can't do the job we're asking them to do, and so there's no reason to have them, or they're not self-tolerant and they're actually going to be dangerous, right? A non-self-tolerant lymphocyte will cause you to have an immune reaction against your own cells, which is really, really, really bad. So just to kind of give you an example here of what this looks like, here we've got our T cell maturing, right? Here we must first be able to recognize we have an APC here, must be able to recognize the MHC class I, right, the self receptor here, and bind to it. So here we have an example of our T cell not being able to do that, right? These 2 different receptors do not match up. Here, on the other hand, we have a successfully completed binding and recognition of an MHC class I. We're good to go, right? We can move on to the next step. Here, we need to make sure that we don't actually bind to the self-antigen. This is bad, right? This is going to cause this T cell, okay, potentially to destroy this cell. Really what we don't want to have happen. So this cell is going to get destroyed. This is the ideal. We bound to the MHC class I, and we're recognizing that this is our cell and we're leaving it the hell alone. That's the ultimate goal here. This is a successfully mature T cell. From there, right, we're going to call them naive, right? Immunocompetent, capable of doing the job, but naive, right? So this is you guys right now, right, until you graduate from college and you go off and actually get some job experience, right? We send you out from school to go off to where you want to go, right? So B cells get exported from the marrow to our lymph nodes. And we talked about before, we have lots of B cells in those follicles within the lymph nodes. T cells get dumped out into the blood to kind of circulate. Eventually, they're going to encounter a foreign antigen. This is the whole reason of their existence, right? At that point, our naive lymphocyte activates. And all that means is that we've just kind of decided that we're going to continue on the life cycle here. We're going to select this on for further development. And that further development is called proliferation. We're going to make exact clones of that exact cell. Okay? And those clones come in two flavors. Effector cells, okay, which are going to actively produce infection-fighting cells, or they're going to be memory cells, which are going to be part of the secondary, tertiary responses. Memory cells and effector T cells, not effector B cells. Effector B cells remain where they're at. But memory cells and effector T cells are in constant circulation, and they are constantly monitoring for subsequent infections. The ability of us to make receptors that are going to be functional against whatever the pathogen happens to be is entirely genetic, right? This is the reason why when Europeans came to this country and they brought smallpox and other sorts of horrible diseases, they were minimally damaging to the European populations, but were devastating to the Native Americans. European populations had lived with smallpox for hundreds of years, if not thousands of years, right? But it was never in the New World. It was never in North America. And so the Native American populations had no capacity to actually generate an immune response against it. So it was very, very devastating. This is why novel diseases, particularly animal-human transition novel diseases, are so very, very dangerous, because we have no capacity to actually deal with it. The average person can make somewhere around 1 billion antigen receptors based upon combinations and recombinations of about 25,000 different genes. So we have the capacity to make a lot of these things. It's just that we don't always make them right off the bat. So the unsung heroes of this whole thing here, the antigen-presenting cells, again, are really the big drivers behind this adaptive immune response, right? What they're going to do, they're going to act very similar to how a phagocyte works. Some actually are phagocytes, but instead of swallowing and destroying the whole thing, they save the antigenic determinant, okay? And they stick that antigenic determinant in an MHC class II complex, right? And they present it, right, to a T cell. So some of these APCs are going to be dendritic cells or macrophages, things that we've talked about before. And dendritic cells and macrophages activate naive T cells. B cells are actually antigen-presenting cells themselves, but they don't activate naive T cells. They, with the help of a kind of a T cell that we're going to call a helper T cell, actually activate themselves. So they bring it to the helper T cell, they activate themselves with it. Activation of T cells is kind of complicated, I guess. It's a two-step process. Basically, we bind to the antigen, right? So the MHC class 1 or 2 or what have you, we bind to it. And then we have a process called co-stimulation, where we release cytokines, right? We've talked about cytokines before, just cell communications chemicals. The specific kind of cytokine here, they're going to be called interleukins, right? Specifically, they're going to communicate between, hence the interleukocytes, hence the leukin. Specifically here for this particular process. And there's a bunch of different kinds of interleukins. We're not going to worry about the different kinds. We're just going to call them all interleukins. As it turns out, there's actually 2 different kinds of naive T cells. Okay, there are CD4 cells and CD8 cells. CD4 cells, when they're activated, become helper T cells. And they activate with just an APC, just one APC, a dendritic cell, macrophage, whatever. A CD8 cell, these are going to become the cytotoxic T cells. And these cytotoxic T cells are the ones that actually go out and kill things, okay? And their activation is a little more complicated. We have to have both an antigen-presenting cell and a helper T cell. And that helper T cell is going to release these interleukins for us. So here is the activation of a CD4 cell. So it's going to be a helper T cell at the end of this. we have our dendritic cell with our class 2 major histocompatibility protein, right? And it's got a little antigenic determinant in it. And we're going to bind to our CD4 cell. And we are going to co-stimulate. We are going to communicate back and forth between these two cells, and we're going to cause this cell to divide and to activate. And so this CD4 cell then becomes a whole bunch of different kinds of cells. Some of them are going to be memory cells for the next time this infection comes around, and some of them are going to be helper T cells. For our CD8 cells, it's a little more complicated, right? We have a class 1 MHC, which our CD8 cell is going to bind to, right? Remember, again, we're looking for self, non-self. And a class 2 MHC protein, which our CD4, okay, is going to bind to, our helper T cell. And so we get all three of these guys hooked up together, okay? The helper T cell, because it's bound now to this APC, releases interleukins, in this case IL-2. I don't care that you remember that, interleukins. But these interleukins then cause the CD8 cell to activate, to proliferate, all of that good stuff. B cells, it's a little bit different. We also are going to require helper T cells here, but remember our B cell is our APC, okay? And so we're going to have release of interleukins from these helper T cells, just like before. We can do this without helper T cells, but it sucks, right? It's a really weak response. It doesn't last very long. It's not really very valuable for us. And it looks, well, the proper response looks like this, right? So our APC/B cell, okay, and our helper T cell binding together, right, at this MHC class 2 protein, right? Again, the only one that's going to bind to an MHC class 1 is the CD8 cell, right, or the cytotoxic T cell. Everything else uses an MHC class 2. The release of interleukins from our helper T cell, in this case it's IL-4. Again, I don't care. Interleukins is fine. That causes the B cell itself to mature. At this point, we've got a whole bunch of these guys, right? A whole bunch of cloned cells. We've proliferated. Most of these clones become cytotoxic T cells or plasma cells, whether in the case of CD8 cells or B cells. Cytotoxic cells, again, remember, attack infected cells directly. Plasma cells exist to make antibodies. The other cells become these memory cells, and this is where immunological memory comes in. The whole idea behind immunological memory is that it's a response, right, that's relatively fast and more powerful the next time you get exposed to it. So with that in mind, then we can divide our adaptive response into primary and secondary divisions, basically. That primary response occurs the first time you get exposed to something, okay? And it's slow, right? We have a lag time of maybe a week, maybe about half a week, 3 to 6, 7 odd days in there before we actually start doing anything. And we're gonna peak out just over a week, right? About 10 days is gonna be the peak response time for that primary response. For a lot of things, this means that we're already getting over it, right? If you have a cold, if you have a non-SARS coronavirus, you're going to be over it within 3 or 4 or 5 days, right? This no longer applies, right? It's not useful to you. If you have a SARS coronavirus, like the one that causes COVID-19 or SARS, right? You might get some benefit out of this because this infection is going to last for a really long time. But again, it's a long time after it's already started, right? It's not great, okay? But the beauty of it comes in the secondary response, right? Here we've got a lag time of hours, right? So the same day, the same half a day, right? You're gonna start to mount this response and you're gonna have a peak response within 2 days. And this response not only is gonna be fast, but it's gonna remain very, very high for a long time, even after the infection has gone, okay? It's a really, really, really powerful response to it. So if we look at what's happening here, just in terms of serum levels of antibodies, here with this light blue line, we've got a response to pathogen A, right? Now your first exposure is going to be here 6 days prior to anything happening. A long lag period. And once it finally does happen, we have a response, but it's not great. And then it's pretty quickly going to diminish. Right? We come back to zero fairly quickly. But a secondary exposure, right? Here we have an immediate response, right? And not only is it an immediate response, but it's massive. We produce a ton of antibody against this stuff, and this antibody persists in your bloodstream for a really long time, right? It's a really, really powerful response to a second incidence of the same pathogen. However, if at the same time that we get re-exposed to A, we also get our first exposure to B, this doesn't help us at all with B. We have a primary response to B, right? So we're still suffering from B for a long time, right? Even as we've cleaned up A. This antibody response is going to be what we refer to as humoral immunity, right? And we acquire humoral immunity 2 different ways, right? We can acquire it actively or passively. Actively is when you make your own antibodies, right? This is obviously the best possible solution because, right, this is going to also produce memory cells. This is going to be the only way that we're going to have a secondary response, is through active acquiring of humoral immunity. And we can do this naturally, right, which is just getting sick, right, which is not the ideal situation. We can also get it artificially, which is, again, preferential. This is what vaccines are all about, okay? You have the dead or severely weakened pathogen. They're almost all exclusively dead. They're just really, really minor, weird cases of attenuated, not dead pathogens. And the idea here is that we get the primary response without actually getting sick, right? Which is really the beauty of the whole situation, that we don't ever have to worry about the primary response because we never had any symptoms. If we're using an attenuated pathogen, a weakened pathogen, right? Again, I want to stress, extremely, extremely rare, okay? It can actually cause you to have the illness because it's not dead, right? It's weak, it's not dead. So not only is this a rare situation to use an attenuated pathogen, it's also extremely rare to get sick from it. So don't worry about getting sick from vaccines. That just is such a minor issue. You can, however, have an allergic reaction. I think we probably all know people that do have allergic reactions to them, not due to the virus within it, but due to the host organism it was grown in. A lot of these are grown in chicken eggs. And if a person has an egg reaction, they can have a reaction to the vaccine because of that. I cannot stress this enough. I know it's 2020. I shouldn't have to say this, but they do not cause autism, okay? It's just not a thing. People are worried about the stuff that's in them. The form of mercury that's in them, most of them don't have it anymore anyway. Even if they do, it's not a toxic form of mercury. Chemicals come in lots of different forms. Please do not worry about vaccines. Please, for the love of anything that you consider good in the universe, please just use vaccines when they're offered to you. They are not bad things. They are extremely good things. We can also acquire humoral immunity passively. This is when we get antibodies from somebody else, okay? Now, the good side here is that this is fast. This is really, really, really fast, okay? But the downside is that we're not making them ourselves. So once the antibodies are gone, our immunity is gone, right? So this is great for us when we're just born, right? Because remember, our immune system isn't active when we're born. There's nothing running, right? And so getting them from mom, either through transmission across the placental barrier, okay, or through breastfeeding post-parturition, this is a way to kind of float us until we can start our own immune system up. We can also artificially acquire them passively through the injection of serum. You may have heard there are some trials in China right now where people are trying to inject antibodies against the SARS coronavirus right now from people that are recovered. Again, this can be wonderful in terms of short-term treatment. It can help people get over something very, very quickly, but it does not give them any sort of memory. Okay, they can, once these antibodies are gone, they can and will become reinfected. So it's not a long-term solution. It's a great term solution in terms of somebody not dying, but it doesn't stop it the next time. Okay, so we'll call it good there. In the next video, we will move on and talk about just a few more things here to do with the immune system, and then we'll move on and talk about the respiratory